Okay, welcome to today's AWRI webinar. The title for today's presentation is Post-pruning and Reworking Management for Reducing Trunk Disease Infection and Spread. I'm very pleased to welcome our speaker, Dr. Regina Baaijens. Regina completed her PhD in plant pathology at Lincoln University, New Zealand in 2011, focusing on the role of nurseries in the spread of trunk disease pathogens in New Zealand vineyards. Reggie's current project is titled Management of Grapevine Trunk Diseases for Vineyard Longevity in Diverse Climates of Australia. This is a collaborative project with SADI, funded by Wine Australia and CSU. So it's fantastic to have Reggie's experience and expertise on hand today. And with that, I'll hand over to Reggie to get us started.
Hello, everyone, and thank you for joining me for today's webinar. This is really a great privilege for me to share some of the main outcomes of my research in the last 5 years, looking at the management of grapevine trunk diseases in Australian vineyards. But for today's webinar, I will focus my talk on post-pruning and reworking management options for reducing trunk disease infection and spread. Now, just to give you a brief background about trunk diseases, I know most of you are already familiar with this, but here in Australia, Eutypa dieback and Botryosphaeria dieback are considered the two most important trunk diseases of grapevines. These diseases are caused by several species of fungi, and these fungi produce spores that infect primarily on pruning wounds, and this can lead to cankers, dieback, and eventually death of vines. Now, since pruning is a standard practice in viticulture, basically we're giving this fungi the opportunity to infect our vines. And this is why they're considered a serious problem of grapevines worldwide. Now, in terms of economic impact, here in Australia, for example, yield losses of up to 1,500 kilograms per hectare have been reported for Shiraz vineyards. And in California, economic losses of up to $260 million per year has also been recorded. And this is why these two diseases are ranked in the top 5 priority diseases by the Australian wine and grape industry. So just to quickly show you some of the symptoms of these trunk diseases. So this is a vine affected by Eutypa dieback. And as you can see, and the cordon or the arm on the right side has died back. But on the left, if you look at this, there's the shoots are severely stunted. And if you look at this closely, the leaves are usually distorted and yellowing, and the internodes are very short. And if these shoots produce berries, they're usually very small and poorly developed. Now, these stunted shoots are due mainly to the phytotoxins produced by the fungus Eutypa lata, which is the primary pathogen causing this Eutypa dieback. The toxins are actually produced by the fungi in the wood, but these are translocated into the foliage, causing this stunted growth. Sometimes these foliar symptoms can be mistaken as herbicide toxicity, and this is how we usually distinguish Eutypa dieback from other trunk diseases in the vineyard. Now, in the case of Botryosphaeria dieback, again, the vine dies back. Here you can see some parts of the cordon or arm have died due to the infection of this fungi. So there's lack of vegetative growth. However, these few shoots here looks normal, and so Botryosphaeria dieback usually do not produce the foliar symptoms that you see with those Eutypa dieback affected vines. Now, both Eutypa and Botryosphaeria dieback can cause cankers on the woody tissues of the vines. So here you can see these vines have severe cankers that goes all the way down to the base of the trunk. And when you scrape this bark, you will usually see this necrotic wood. And when you cut this wood cross-section, you will usually see this wedge-shaped necrosis. And this kind of wood symptoms are associated with both Eutypa and Botryosphaeria dieback. Occasionally, you will also see this central staining of the wood. And these kind of symptoms are usually just associated with Botryosphaeria dieback. So since these two diseases are considered a threat to the sustainability in Australian vineyards, Wine Australia basically funded this research looking at management of these trunk diseases in Australian vineyards. And this project is a formal collaboration between SARDI and the National Wine and Grape Industry Center at Charles Sturt University, as well as in collaboration with University of Adelaide. And Mark Sosnowski is our project leader. And while Sandra Savoca is my project supervisor here at NWGIC. Now for today's webinar, I'll focus my talk on the 4 objectives. So one is looking at the spore dispersal patterns of these pathogens. And we also look at the pruning wound susceptibility to these spores and how to protect these pruning wounds from infection. And lastly, looking at remedial surgery as a management option for vines that were affected with Botryosphaeria dieback. With the spore trapping, before we started with this project, there are no data available on the spore dispersal patterns of this Eutypa and Botryosphaeria dieback available for Australian vineyards. There are some studies done overseas, including California, Europe, as well as South Africa, but some of these data have conflicting results. So there is a need for us to collect our own local data. So starting in 2013, we did the spore trapping at the 4 different climatic regions. So 2 spore traps were deployed in South Australia, one at the Barossa Valley and one in Coonawarra. And in New South Wales, we also deployed 2 spore traps, one at Hunter Valley and one in Griffith. The objective of this work is to investigate the spore dispersal patterns of these Eutypa and Botryosphaeria dieback pathogens and determine the climatic conditions or environmental factors that triggers the release of the spores. And it will hopefully help us identify the high-risk infection periods in Australian vineyards. Now, as part of this work, we also developed DNA-based tools that we use to analyze the spore trap samples. Prior to this project, there's no DNA-based tools available that we can use, so we developed this quantitative PCR that we use to detect and quantify these spores, since DNA-based tools are shown to be highly sensitive as well as more accurate compared to the conventional techniques. So today, I'll just present the 1-year data that we collected from each location. The graphs on the left are those from South Australia, and the ones on the right from New South Wales. So just very quickly, I'll just explain what this graph means. So these green lines here represents maximum temperature. These red bars represents Botryosphaeria dieback spores. These dark gray bars represents Eutypa dieback spores, and these blue peaks represents rainfall. And here, just by looking at these graphs, it's showing that Eutypa dieback and Botryosphaeria dieback spores are released sporadically at different times of the year. And we also found that that spore release differs between region. Particularly in South Australia, we found that the spores were released mostly during winter, particularly in Coonawarra, as you can see in July and August. However, there was less spores detected during summer for both Barossa and Coonawarra, especially in January 2014 and February 2015. In contrast, we found a high number of spores were trapped during summer in both Griffith as well as Hunter Valley, and less spores were trapped during winter for these 2 regions. So just to summarize these results, we found that the seasonal release and the number of spores differed between regions, wherein we trapped a high number of spores in winter in South Australia, whereas in New South Wales, we also trapped a high number of spores during summer. Now, these differences in regions were mainly attributed to rainfall. We found that rain is a very important factor in the spore release, wherein we were able to trap spores during or immediately after the rain. And what we found interesting is that the amount of rain was not correlated with the amount of spores. So whether you get 1 mil of rain or 100 mils of rain, we were able to detect these spores. However, it's also important to know that not all rain events resulted in the release of the spores. And the amount of time for the fungi to produce new spores may explain this lack of spores trapped during some rainy periods. And also on a few occasions, spores were trapped more than a week after a rain event. And the spore release during these dry periods may be attributed to other environmental factors like relative humidity or dew. But just based on this data, we found that the Eutypa and Botryosphaeria spores are released in the vineyards all year round. So these spores can basically potentially infect any pruning wounds that you make on your vines. So as part of our work, we also look at pruning wound susceptibility to determine how long these pruning wounds are susceptible to these spores. We also look at the optimum pruning time, whether if you do your pruning early or delay it in winter, whether it will reduce infections. So we look at 3 pruning times, which is early pruning that was done in June, mid pruning in July, and late pruning in August. And after each pruning, these pruning wounds were inoculated with either Eutypa spores or Botryosphaeria spores. So the inoculation was done 1 day, 7 days, 14 days, up to 112 days after pruning. And the following year, those pruning wounds were assessed for the presence or absence of the pathogen. Now, I'll just present the summary of the data that we got from this 3-year field trial. And this is for Eutypa lata. So just to quickly explain these graphs, those bars represents the number of pruning wounds or the percent pruning wounds where we were able to recover Eutypa lata. And here you can see that very first day after pruning, whether you do your pruning early, mid, or late, those pruning wounds are highly susceptible to these pathogens. However, as the pruning wound starts to age or they start to basically heal, they become less susceptible to these spores. However, we found that even up to 14 days, they can be potentially infected by these Eutypa lata spores, and therefore it's important to protect those wounds from getting infected. In the case of Botryosphaeria dieback, again, we got some similar results. For the second graph, we found that, with Neophococcus luteum, again, whether you do your pruning early, mid, or late, that very first day after pruning, when the wounds are still fresh, they're really highly susceptible to these spores. However, as the pruning wound starts to heal or age, they become less susceptible. But in the case of this Neophococcus luteum spores, we found that even up to 28 days, those pruning wounds were infected. And this is mainly because we use a high dose of spores when we did the inoculation. So to further confirm these results, we repeated this trial, this time using Diplodia seriata, which is another species causing Botryosphaeria dieback. And this is the most prevalent species actually present here in Australia. And this time we reduced the dosage to really simulate what's happening in the vineyard. And here you can see that after 14 days, these pruning wounds are susceptible to these Diplodia seriata spores. But after that, they're not susceptible to these spores anymore. So based on this data, we found that these wounds are basically most susceptible during the first 2 weeks after pruning. However, as the pruning wound starts to heal or age, they become really less susceptible to these spores. So therefore, there's also a need for us to look at pruning wound protection to make sure that those pruning wounds can be protected during the first 2 weeks when they're most susceptible to the spores. So we look at 3 different products that are commercially available for viticulture here in Australia. So this is Folicure, which is with the active ingredient tebuconazole, Cabrio, which is pyroclostrobin, and Emblem, which is fluacinum. Now, prior to testing these products in the field, these were actually previously tested in the lab with the other fungicides. And, so we did some in vitro testing, and based on that test, we found that these 3 are the most promising, and that's why we further tested them in the field. We look at 3 different, 2 timing of application. One is curative, wherein we did the pruning, and then we inoculated these pruning wounds with either Eutypa or Botryosphaeria spores. And the fungicides were applied at 1 day, 3 days, and 6 days after inoculation. We also look at preventative application where we did the pruning and we applied the fungicide straight away. And then the inoculation was done 1 day, 6 days, and 14 days after we applied the fungicides. And again, those pruning wounds were assessed for the presence or absence of the pathogen the following winter. So today, again, I'll present the summary of these results that we did for 3 years. And this is for Eutypa dieback. And just very briefly, just to explain this graph here, these dark gray bars, these are the pruning wounds that were inoculated with Eutypa spores and were not treated with fungicides. And as you can see, if you don't apply anything, you will get around 60% infection. However, if you apply the fungicides the day after infection or 3 days and even up to 6 days, you can significantly reduce that infection. And the good news is the same fungicides work the same way with Botryosphaeria dieback. So it's like hitting two birds with one stone. So again, if you don't apply any fungicides and there's spores present in the vineyard, you will get around 80% infection. However, if you apply the fungicides on the day after infection, as well as 3 days or 6 days, you will significantly reduce that infection. However, if you delay the application up to 6 days, the results are not as good as if you apply them early. But based on these results, we can summarize that all three products reduced Eutypa dieback and Botryosphaeria dieback infections, and the curative effect was up to 6 days after infection. Now for preventative application, again, we found that these products really work. So here again on this graph, so these are the pruning wounds that were not treated with fungicides. And here you can see if you don't apply any preventative fungicides, then you will expect around 60% infection. However, if you apply that right after pruning, then you will significantly reduce that infection. However, here, if you look at here at 14 days, even those pruning wounds that were not treated with fungicide, after, at 14 days, there was less infection, and this is mainly due to the pruning, the healing of the pruning wounds or the aging of the pruning wound that's actually preventing the fungi from infecting those pruning wounds. And these results also confirmed the pruning wound susceptibility work that we did, that those pruning wounds are only highly susceptible to these spores on that first 14 days after pruning. And again, we got similar results with Botryosphaeria dieback. And here again in the graph, it's showing you that if you don't apply any preventative fungicides, you will expect around 80% infection. But if you apply those fungicides, you will significantly reduce that infection up to 14 days. So again, to summarize, all these three products protected those pruning wounds against Botryosphaeria dieback and Eutypa dieback infections, and the preventative effect is 14 days after application. So basically, based on this data that we got, we've shown that there's a 6 days curative effect and 14 days preventative effect. So you have that sort of 3 weeks window where you can actually apply those fungicides. So you don't need to rush when you're busy during the pruning season, because you have that 3 weeks basically to apply those fungicides. And as we know now, we've shown you that those pruning wounds are only susceptible to these spores on that first 2 weeks after pruning. So just to quickly refresh your memory, these are the products that are currently registered for managing Eutypa dieback here in Australia. So Garrison, as well as the Vinebacks, are actually vine wound dressing, whereas the fluoxetinum and the tebuconazole are the ones that you can use for spraying. So these are the ones that we tested in the field. Currently, there are no fungicide registered to manage Botryosphaeria dieback. But since these products works for both Eutypa and Botryosphaeria dieback, again, it's like killing two birds with one stone. The Cabrio that we tested is currently not registered for Eutypa and Botryosphaeria dieback, but we're hoping that we can expand this label. But the current recommendation is to just do one spray during dormancy or during the pruning season, since we have shown that it's got this curative effect as well as preventative effect. So one spray should be sufficient to protect those pruning wounds from getting infected. Now, the last topic that I'm going to talk for today is the remedial surgery. Now, remedial surgery or trunk renewal has long been used to control Eutypa dieback. So the current recommendation suggests to cut all the infected wood and an additional 10 to 20 centimeters of the non-symptomatic wood. And once you get the water shoots, these water shoots can be trained to replace the missing vine or cordon. However, this current management practice was recommended for Eutypa dieback. So there is a need for us to investigate whether this remedial surgery could also be used to manage Botryosphaeria dieback, although theoretically it should work the same way. So to do this, we conducted 3 field trials in 3 commercial vineyards, wherein we cut the vines in 3 different heights. So for high cut, basically we cut the trunk just below the crown. Mid-cut was midpoint between the ground and the crown, and low cut is 20 centimeters above the graft union or 20 centimeters above the ground. And we want to see the effect of this cut on the shoot production as well as the recovery of the vines from remedial surgery. So as I mentioned earlier, we did this trial in 3 commercial vineyards, and prior to remedial surgery, we look at the disease severity of all these vines. So for vineyard 1, these are 24-year-old grafted Semillon. Vineyard 2 is a 20-year-old own-rooted Semillon, and vineyard 3 is a 24-year-old own-rooted Shiraz. The disease severity at the time of the surgery differed between the 3 vineyards, with vineyard 1 being the most severe at around 75% dieback severity, and vineyard 3 being the least severe of the 3 vineyards, wherein the dieback severity was only around 35%. After remedial surgery, we monitored the shoot production for 3 years. And what we found is that for vineyard 2 and vineyard 3, they recovered really well, and all vines produced new shoots, regardless if we cut them low, mid, or high, as you can see here on the graph. However, for grafted vines, particularly those that were cut low and near the graft union, they tend to produce shoots from the rootstocks rather than from the scions, as you can see here in the photo. And these results basically suggested that remedial surgery may be more suitable for own-rooted vines rather than grafted. However, we tried to repeat this, so we're still doing another trial using grafted vines to further confirm these results. But based on this preliminary data that we got, it is probably necessary to consider other options, like, for example, regrafting or tap working to manage these trunk diseases on grafted vines. Now, what happens if you don't do any reworking? So these are the vines that we monitored. These were not reworked, and we monitored the dieback severity for 3 years. And what we found is that we observed 5 to 10% increase of dieback severity each year. And basically, this is telling us that if you don't apply any interventions, this can lead to the death of vines. So if you don't do anything, eventually your vines will die, particularly those who are already severely infected. Now, just to quickly show you some of the vines that we reworked. So on the left is a vine that we reworked in November 2015. And on the right, this is what the vines look like 2 years after. So you can see there's already a nice new trunk and cordon, and the growers were already able to harvest some berries from these vines. So the vines recovered really well. However, the following year when we visited this vineyard, we already started to observe new dieback symptoms. The severity is still relatively low, to up to 3%. However, the message here is that even if you do reworking, but you don't apply any pruning wound protections after reworking, these vines will eventually get new infection. So it's like going around in circle. So it's really important to be proactive, that after reworking your vines, make sure those new pruning wounds are protected. Otherwise, they will eventually get new infections. So we're continuing with the monitoring of these vines until next year, and even hopefully extend it to another 2 years. But basically the message here is that make sure those pruning wounds are protected, otherwise they will just get new infections. Now in 2017, we further got funding from Wine Australia to continue with this project. And now we have expanded our spore dispersal surveillance to 6 sites. We're also currently looking at infection thresholds of nursery plant materials to see whether these contaminations from the nursery plant materials may actually contribute to the infection in the vineyard, since there's no data available in Australia about these nursery plant materials. And as I mentioned previously, we're continuing with the remedial surgery for grafted vines to further look at the effect of this remedial surgery on shoot production, as well as how to improve shoot production for these grafted vines. And lastly, we're also developing these in-field DNA-based diagnostic tools we call LAMP. And the purpose for this lamp is something that the industry can use in the field, so that you can do your own testing as well as do your own spore trapping monitoring, for example. But at the moment, this is still in a very early stage of development, but hopefully this is something that we can recommend to the industry in the future. So thank you very much for your attention. I also want to thank our collaborators as well as industry partners for supporting our project.